Hi, I'm Parker.
I'm Tianjun.
Hi, I'm Dario.
All right, and today we are going to be talking to you guys a bit about tagging myelins with green fluorescent proteins inside of the brain of patients with demyelinating and degenerative brain diseases. Okay, so let's see. Myelin is a protein that forms inside of the brains of most living organisms around the nerve cells, specifically the axons, which are the main communication systems between each and every neuron inside of the brain, and it's essentially what creates a functioning circuit which delivers electrons. And in order to move these electrons as effectively as possible, say when you are practicing like a dance move or recalling information, these electrons will travel through the axons, and as you become better and better at doing such things, the more protein will be established and grown around the axons, which is essentially this insulating protein. It's an insulator that helps retain as many electrons and keep the flow going as strongly as possible. However, there are many diseases and many neuronal conditions which can unfortunately break down and stop the processes that help create myelins around the axons, which including Parkinson's or multiple sclerosis, and this demyelinating process can cause motor issues, issues regarding the retrieval of memories, and it can be generally bad for the sufferer or patient. And so we believe that we can actually next. So our concept is to tag these neurons with green fluorescent proteins within their own genetic code, and to essentially try to locate and study each individual neuron over a set period of time, and also being able to study it under certain conditions, while also being able to observe and image it through different methods of fluorescence microscopy and spectroscopy while the patient or subject specimen is still alive. And you wouldn't really need to remove any brain cells necessarily to observe them and how they are affected by these diseases. Okay, so well, many people are affected by these diseases, such as Parkinson's and multiple sclerosis, and every year there are thousands of people who are diagnosed, and generally that is devastating news, and it can it's just generally very bad. And as it continues to spread and attack the brain, it can cause degradation of memory and behavioral. Okay, so, but we believe that by, again, tagging the genetic code of the neurons with green fluorescent proteins, we can hopefully better understand the complexity of how the brain reacts to these diseases and how it's affected. And we believe that these studies could help us hopefully one day create a cure or prevention method from actually getting these diseases.
So common studies have been done on zebrafish and mice. So these experiments were conducted 6 months ago, and they were exposed to a myelin-related disease called demyelination, as he mentioned earlier. And scientists studied the effect of the GFP-tagged myelin and in these organisms and found that the diseases were easier to see, and they could understand neurological concepts and mapping the brain in an easier way. Okay. So our proposed project is to essentially tag the genetic code of neurons in the brains of the demyelinating diseases suffering patients with green fluorescent proteins. By using GFP tagged myelin, we can have the ability to trap individual neurons and study them over a certain period and examine the brain for further details that might help us determine how the disease is caused and how it affects our brains, and find ways to prevent and cure myelin-related diseases.
So along with our proposal, there are potential challenges that will arise that we have to take into account. Well, since the GFP is essentially a guide, and since they guide by providing a visible glow, and it will be really difficult to monitor the GFP inside a deep inside the tissue of a brain. And to do that, well, there are a variety of ways to actually do the imaging. Well, what we've learned over the course of these days are there are the scanning probe microscopy, electron microscopy, fluorescence microscopy. But then, even though there's these new and improved methods of imaging, it is still really difficult to see inside the deep tissue of the brain, because if we can do that, then technically we wouldn't be here. And also, since we have to introduce the modified nerve cells into the affected victims of the disease, we have to technically extract some healthy cells from them to actually avoid collisions. Well, not collisions of the modified cells. We have to prevent the potential invasion. So this method will be really personal, based on the patient himself or herself. So we can't mass produce this potential cure. And so overall, the GFP-tagged myelin proteins could possibly allow many scientists to study the complexity of the neurological systems and to develop treatments and cures to brain-related diseases with fluorescence microscopy. And with this discovery, we can look at the brain diseases on a whole new viewpoint. And the potential future, in the near future, we can potentially be able to track back the diseased brain cells to find the origin of the disease and potentially to solve the problem. And here are the contributions and references.
Any questions? yeah. Any questions?
One question. In the idea of removing nerve cells, what would be one of the major problems you would encounter? You're trying to treat a patient whose nerves are suffering from disease. So in removing those cells, what problems do you think you would encounter?
Okay, so, well, the thing about these nerve cells, these neuronal cells inside the brain, as they're being attacked by these degenerative diseases, there is a very high risk of them ultimately being destroyed or killed off by said disease. And of course, there's also ways that these cells will, not be able to fully connect and be integrated into the rest of the brain. And, that's because the nerve cells, they need to be connected by the axons. However, there are lots of different methods being created to fuse cells, nerve cells, neuronal cells together to one another in order to create an established connection. But, again, as the cells are being attacked, it's very risky, and, for said cells. That's why you need to find a lot of them, and you need to find a lot of younger cells that might also be healthier, perhaps say from a donor or a stem cell donor that could potentially help grow these brain cells. There's also lots of other methods of creating brain cells through stem cells in labs, or essentially mini brains, as they call them. But these stem cells would essentially, again, be tagged with a GFP so that we can track them as they divide throughout the brain, or probably, this way we can actually track specific neurons while also trying to
find the causes of the diseases.
Yeah.
So let's say if you take the neurons out, you're going to immediately cause the damage that you're trying to prevent.
Exactly.
Right?
Yeah.
However, you're really lucky. Are you aware of which type of cells actually make myelin? So it's a neuronal cell.
Yeah.
It's not neurons.
Yeah.
It's glial cells.
Yeah.
And it turns out
The
that
glial cells.
you guys are in one of the few areas. They are removing glial cells from patients, growing them up in batch cultures, and re-injecting them into patients right now. It sounds like it's sci-fi that you would ever be able to do this.
Yeah.
And you can't do it with neurons. That's
Exactly. Yeah. No.
out,
Yeah.
right? But you could add a GFP tag to those. So then the next question I have is, what are you going to learn by studying humans that you haven't already been able to learn from rodents and mice and these other models?
Well, let's see. So,
well, maybe we'll have the kind of a safeguard that this worked on humans. It worked on one, maybe it will work on the others. And with a mouse or rodent, it technically is it could create some trusting issues.
So like validate the research from mice.
Yeah, potentially.
and humans have much larger brains than mice, and so there's lots of different areas of the brain for which these different diseases could attack and target specifically. I think that by studying how it works in larger brains of larger organisms, we could better study exactly how and why this disease does, what it does, and in these different areas of the brain. And I think that although mice, are a good test subject to use for these methods of disease prevention or repair, human brains, they could also provide a much larger area to work with and to better understand exactly how these diseases, can affect a brain based upon exactly where, it again does pinpoint and focus its areas of, damage. Like where these, you get it. Yeah.
So I think we have time for one more question. I think, Dr. Steve, you had a question?
Yeah, I was just going to ask, you mentioned that, this point here on the top, I'm going to just ask you again, similar to the last group. Did you hear anything this week that would allow you to see deeper into the brain? Right? I know that you did, so I'm just wondering if you thought about other methods that would allow you to basically look at demyelination using fluorescence microscopy throughout a brain rather than by extracting cells,
Yeah.
and what the limitations of those approaches might be.
Yes. Well, again, there are many different types of fluorescence methods through microscopy. There's, of course, removing brain samples or tissue samples that have been tagged with the GFP or other fluorophore proteins and markers. And, that's great and all. You can see the individual cells up close and in a very clear image. However, of course, through doing that, you can either end up damaging the live specimen or injuring it or ultimately killing it if you are removing brain cells. But there are other methods of microscopy, fluorescence microscopy, that can be used to observe the live samples or specimens without actually causing damage to them, such as, if I'm correct, MS spectroscopy, which is well, yeah. There's many types of scanning machines which can essentially highlight and observe the fluorescent reactive proteins or materials that can be found within a live specimen fully through methods of scanning. We talked about it a little bit while we were doing the tours and talking about methods of fluorescence microscopy and using live mice, specifically more or less ones that have had quantum dots injected into their bodies. But of course, it's also possible to use other types of fluorophores that might be more, might be easier to work with and easier to observe.